This is the fourth and final lesson of Chapter 6. We are going to continue talking about enzymes, which recall from the previous lesson, enzymes are biological catalysts that increase the rate of chemical reactions. So we're going to start with this lesson by looking at some factors that might affect the rate of the reaction, specifically in how they affect the enzyme and its ability to do its job. One factor that will affect the rate of reaction is the availability of the substrate that the enzyme is binding to. So first, let's look at the graph on the left, which is showing you how the rate of a reaction is affected by the substrate concentration, which is on the x-axis. So notice that there's a pretty steady increase in the rate of the reaction as the amount of substrate is added until there reaches this point, this X, green X, called the point of saturation. And after that point, even if you continue to add substrate, there's no increase or decrease in the rate of the reaction. It's called saturated. Point of saturation means that, well, what's really happening is if you look at the illustration on the right, there comes a point where you can continue adding substrates and there's enzymes still available to bind those substrates to catalyze the reaction. But if there are too many substrates and not enough enzymes to bind them, like in the middle scenario here, you see a few extra substrates hanging around. Now those original ones will probably be changed to the products pretty soon, and then the available substrates will bind to the enzymes. And so that will probably still increase the rate of the reaction, but in the third and final scenario, there's way too many substrates there and not enough enzymes, so it doesn't do any good to continue adding more substrate after that point of saturation. In fact, if you wanted to increase the rate of the reaction, what you would have to do is add more enzyme. So this is one factor that can influence the rate of a reaction, even with the presence of an enzyme. Another several others, but one example of one here are the environmental factors. So one environmental factor is temperature. Recall that enzymes are proteins, and proteins their function is determined by their shape. And if the conditions in the environment are not really optimal, specifically temperature or other things like pH, salinity, or a few other examples of environmental conditions, they can actually denature or change the shape of the enzyme. And with that, it wouldn't be able to do its job. So as shown here, we're comparing in a different graph the effect of temperature on the rate of reaction in two different organisms. One on the left in the black graph is showing the optimal temperature for typical human enzymes. So you can see that in general, our body enzymes work best at about 37 degrees Celsius, where the peak is. And there's a pretty steep decline in the rate of reactions once we go beyond 37 degrees. And similarly, the same trend is seen for the thermophilic heat-loving bacteria, except the whole graph is kind of shifted to start at 40 degrees Celsius, which is pretty high for us and perhaps fatal for the human body. But then their optimal temperature is about 75 degrees Celsius. And then again, after that point, when it's hotter, too hot, there'll be a decline and the bacteria can't even survive really beyond 80, 90 so degrees Celsius. So again, there's several factors that can affect the rate of the enzymes, the rate of the reaction that is catalyzed by an enzyme, including substrate concentration, enzyme concentration, and environmental factors such as temperature. So when we're looking at, go back to this sort of organizational chart here to just help us summarize some of the key points about enzymes and the types of enzymes that play roles in chemical reactions. That's what this is showing. At the top, we just talked about how enzymes speed up the rate of chemical reactions and what can affect their rates. Now, let's drop down to look at the different types of enzymes, those that are inhibitory and those that need to be activated. And then we'll talk about the subtypes of those and look at some examples. So I want to start, before I get into the more general types, with an example of where enzymes are important in a physiological process in an organism. This very probably scary, intimidating diagram is the citric acid cycle that we'll study when we talk about cellular respiration very soon. And right now, I don't want you to panic about memorizing the diagram. In fact, you won't even have to for cellular respiration. You'll have to know certain key points of it. But for now, I just want you to focus on the words that are in blue text. These are all, most of them at least, except water, CO2. Everything else, though, is an enzyme. And so one thing I want you to take note of with those enzymes is the way they're named. For example, citrate synthase. Citrate synthase, I know that's an enzyme because typically they're named with a word that ends in ASE. So if you look around, you might see that same thing for the ones that are fully listed here: succinate thiokinase, succinate dehydrogenase, fumarase. This is an abbreviation for something. But in general, you see that ASE. So that's a good clue to know that that's an enzyme. Enzymes are also typically named after the substrate that they bind to. So acetyl coenzyme A is a substrate that binds to the enzyme citrate synthase. So there's some relationship there with the way that they're named. Again, I just wanted to kind of point out an example of this and also to show how important enzymes are. Each step of the citric acid cycle is catalyzed by an enzyme. So if something were to happen to one of those enzymes where it didn't work as it should, perhaps like the temperature is not ideal or there's not enough substrate there to saturate the enzyme, then that could affect the whole cycle and ultimately the ability of that organism to make ATP, because this is just one piece of that process that leads to the synthesis of ATP. Now, in this cycle, some of these enzymes are not always available. They might need to be produced by the cell as needed, or they need to be perhaps turned on to be activated. So this brings us to the two ways that enzymes can be turned on or made available in a chemical reaction. One way is that they're only produced as needed. So, perhaps the cell doesn't have that enzyme readily available, but through gene regulation, through its genetic code, in a particular stage of its life cycle, perhaps it's going to start producing a specific enzyme when needed, instead of just wasting energy and materials to make something that it doesn't actually need all the time. And the second way is to activate the enzyme. So maybe it's there and it's kind of just, sitting around waiting for its turn, but it has to be turned on, activated. And to activate, something is usually added to make the enzyme work, another component. These can be two different types of molecules. Cofactors are non-proteins. Usually they're inorganic, such as metals. One example here is shown. This is hemoglobin, which is an enzyme that facilitates the binding of oxygen and red blood cells throughout our body. So notice that in the hemoglobin, there's a lot of different molecules there, including different polypeptides shown in the different colors. When you see like alpha helices, beta pleated sheets, but focus on the things that are labeled there, like the iron sulfur clusters. So that's a cofactor. Heme is another cofactor. In addition to the cofactors, hemoglobin also has coenzymes, which are organic molecules that are added to the enzyme to activate it. So these might be things like vitamins. So for heme here, or I'm sorry, for hemoglobin, we have heme. I'm sorry, no, that was actually a cofactor. Just kidding. The ones that show the molecules, those are carbons, the gray molecules. So quinone and FAD are the coenzymes for hemoglobin. You won't have to memorize that as an example, but definitely make sure you understand that there's two different kinds of molecules that can be added to the enzymes and how they differ, but they both serve the same function to activate enzymes. Okay, now on the flip side, sometimes, if I go back to the summary chart here, as a reminder here, sometimes enzymes need to be inhibited or turned off, let's say, or prevented from being able to do their job. So they might be there in the system and cell, but they're not meant to be always working, always doing their reaction that they catalyze. So there's two different ways that these enzymes can be inhibited. One is competitive inhibition, and one is allosteric or non-competitive inhibition. So let's zoom ahead to that. And so here is a diagram on the left showing the two different types. Competitive inhibition is when you have a different molecule that can bind to the active site, preventing the substrate from binding to the active site. That competitive inhibitor kind of tricks the enzyme into thinking it is the substrate. Even though it's not exactly the same in terms of its structure, as long as it binds to part of the active site, it can still block the substrate. So with that, the enzyme can't really do its normal job releasing the substrates that it would if the, releasing the products that it would if the substrate were to bind. The other scenario on the right is non-competitive inhibition. Notice that the substrate in turquoise here at the top can still find the active site, but it's actually not going to fit properly because a non-competitive inhibitor has found a different point of attachment. And when it binds to the enzyme, it actually changes the shape of the enzyme so that no longer will it bind to the substrate that it should. So another term for non-competitive inhibition is allosteric inhibition because the site where the non-competitive inhibitor binds is also known as the allosteric site. So be sure you understand the different terminologies that are used in these scenarios. And with that, we've finished the lesson on enzymes and also all of the lessons for Chapter 6.